Before the 1980s, signal transduction pathways remained partially uncharted territory, particularly concerning vasodilation and the intricate mechanics of smooth muscle relaxation. Robert Furchgott, Louis Ignarro, and Ferid Murad resolutely confronted a significant enigma: endothelium-dependent relaxation, a phenomenon that hinged on an elusive endothelium-derived relaxing factor, or EDRF. Their combined efforts revealed that nitric oxide, or a closely related nitroso compound, fulfilled the role of EDRF. Furthermore, they demonstrated that nitric oxide activates guanylate cyclase, an enzyme that catalyzes conversion of guanosine triphosphate to cyclic guanosine monophosphate, cyclic GMP, a pivotal mediator of smooth muscle relaxation. Furchgott's initial experiments provided foundational evidence. He observed that acetylcholine induced relaxation of blood vessels only when endothelial cells lining the vessel walls remained intact. His meticulous work strongly suggested the existence of a signaling molecule produced by these endothelial cells, a molecule responsible for triggering smooth muscle relaxation. Hence, endothelial cells were indispensable for the vasodilatory effect of acetylcholine. Endothelium-dependent relaxation, as it became known, intrigued researchers and stimulated a concentrated pursuit for the underlying mechanisms. Building upon Furchgott's assertions, Ignarro conducted a series of experiments indicating that EDRF was either nitric oxide itself or a closely related compound containing the nitroso group. Through rigorous pharmacological and chemical analyses, he found that nitric oxide mimicked the effects of EDRF, triggering smooth muscle relaxation in a remarkably similar fashion. His claims rely heavily on the use of biosacascades, where the effects of EDRF released from endothelial cells were compared directly with those of chemically synthesized nitric oxide. Spectroscopic analysis further bolstered his findings, revealing comparable signatures for both substances. Concurrently, Murad's research illuminated the intracellular signaling pathway downstream of nitric oxide. He discovered that nitric oxide directly activates guanylate cyclase, the enzyme responsible for synthesizing cyclic GMP. This activation led to an increase in intracellular cyclic GMP levels, subsequently causing smooth muscle relaxation. Murad's experiments highlighted a previously unappreciated mechanism by which a gaseous molecule could regulate cellular function via cyclic nucleotide signaling. Initially, this concept faced skepticism due to the prevailing notion of nitric oxide solely as a toxic pollutant. The experiments supporting these claims were diverse and compelling. Furchgott utilized isolated blood vessel preparations to meticulously observe the effects of acetylcholine under varying conditions. Ignarro employed sophisticated chemical techniques and biosacascades to compare EDRF with nitric oxide. Murad relied on enzyme assays and cell-based studies to demonstrate nitric oxide's activation of guanylate cyclase. The evidence accumulated through these distinct yet interconnected studies provided a formidable case for nitric oxide as a crucial signaling molecule. Before this revolutionary work, nitric oxide held recognition primarily as an environmental pollutant stemming from combustion processes. It was perceived as a toxic byproduct rather than a biological messenger. The prevailing view placed emphasis on traditional neurotransmitters and hormones mediating cell-to-cell communication. The idea that a simple gas like nitric oxide could possess potent signaling capabilities challenged existing paradigms, shaking the foundations of cell biology and pharmacology. Even prior to this transformative research, scientists recognized the vasodilatory effects of certain compounds, such as acetylcholine. Studies had shown acetylcholine's ability to relax blood vessels, though the precise mechanism remained elusive. The discovery of endothelium-dependent relaxation elucidated this mechanism, revealing the crucial role of endothelial cells in mediating acetylcholine's effects via nitric oxide production. Nitroglycerin, used for treating angina, provided another clue. Although physicians understood its therapeutic benefits, the underlying mechanism linking vasodilation to nitric oxide generation remained unveiled until the groundbreaking work of Furchgott, Ignarro, and Murad. The legacy of their discoveries extends far beyond the cardiovascular system. Modern research reveals nitric oxide's implications in a multitude of physiological processes and pathological conditions, from immune responses and neurotransmission to cancer development and erectile function. Nitric oxide has emerged as a versatile signaling molecule with far-reaching consequences. Therapies targeting nitric oxide signaling have revolutionized the treatment of conditions like pulmonary, hypertension and erectile dysfunction, underscoring the clinical significance of this fundamental discovery. This essay delves into the groundbreaking research of Furchgott, Ignarro, and Murad. It details their individual pathways and their collective triumph in upending scientific dogma. It surveys individual contributions, examines the scientific obstacles surmounted, and highlights the transformative impact of their findings on cellular signaling and human physiology. Previously known as a toxic pollutant, nitric oxide was revealed as a critical signaling molecule within the cardiovascular system and beyond. The long shadow of Furchgott, Ignarro, and Murad's work extends far beyond its immediate implications within the cardiovascular system. The discovery catalyzed an explosion of research into nitric oxide's multifaceted roles across an array of biological systems. Its impact on fields like immunology, neuroscience, and cancer biology redefined our understanding of cellular communication and disease pathogenesis. In immunology, nitric oxide quickly emerged as a critical mediator of macrophage function, specifically in the context of innate immunity. Researchers soon elucidated the mechanisms by which macrophages, crucial immune cells, utilize nitric oxide to destroy pathogens and combat infections. The discovery that macrophages could produce nitric oxide, catalyzed by the enzyme inducible nitric oxide synthase or INOS, provided a direct mechanism for the non-specific killing of bacteria, viruses, and other invading organisms. This understanding revolutionized our approach to understanding immune responses and the mechanisms of inflammation. The precise techniques employed to study nitric oxide's effects on macrophages included the use of electron paramagnetic resonance, or EPR spectroscopy. This technique allowed researchers to directly measure the presence and quantity of nitric oxide radicals, particularly in biological tissues. Chemiluminescence assays, another key methodology, were employed to detect nitric oxide production by immune cells with exquisite sensitivity, offering a quantitative measure of nitric oxide's synthesis dependent upon enzyme activity. This, in turn, provided insight into cellular responses. In essence, nitric oxide acts as a potent weapon, contributing to the complex orchestration of the immune response. Its presence within the nervous system was a second area that galvanized interest. Nitric oxide functions as a novel neurotransmitter, involved in various neural processes. This contrasts with classical neurotransmitters like serotonin or dopamine. Nitric oxide, a gas, diffuses across cell membranes without the need for specialized receptors or transporters. Its synthesis, within neurons, mediated by neuronal nitric oxide synthase, or NNOS, influenced synaptic plasticity, as well as processes fundamental to learning and memory. The ability of nitric oxide to act as a retrograde messenger, moving from the postsynaptic to the presynaptic neuron, revolutionized the understanding of synaptic transmission. It provided a mechanism for strengthening or weakening synaptic connections, offering the cellular basis for learning and memory. Its involvement in neurodegenerative diseases, such as Alzheimer's disease, added another layer of complexity to its role. Subsequent research, particularly focused on the therapeutic potential of manipulating NO levels within the brain, remains an active area of investigation. The role of nitric oxide in cancer biology presented another surprising revelation. The molecule wields paradoxical influences in tumorigenesis and tumor progression. Research demonstrated that nitric oxide can exhibit both pro- and anti-tumor effects dependent on the cellular context. In some cases, nitric oxide can induce apoptosis or programmed cell death in cancer cells. In others, it can promote angiogenesis, the formation of new blood vessels, which fuels tumor growth and metastases. The development of the novel blood vessels, providing the nutrients to promote tumor growth, is a phenomenon often observed. In this context, nitric oxide can function as a double-edged sword, potentially enhancing tumor aggression. Nitric oxide's impact on tumor immunology, particularly in how it affects immune cells within the tumor microenvironment, and its influence on the sensitivity of cancer cells to chemotherapy, are aspects that continue to capture the attention of cancer biologists. The initial findings of Furchgott, Ignarro, and Murad had an immediate impact on clinical therapeutics. The development of sildenafil, a drug that enhances the effects of nitric oxide, provided a revolutionary treatment option for pulmonary hypertension. It relaxes the pulmonary vasculature, reducing its pressure and improving circulation. Sildenafil's success in treating erectile dysfunction also highlighted the importance of nitric oxide in regulating smooth muscle relaxation. This unexpected pharmaceutical development offered new therapeutic options. The development of sildenafil revolutionized treatments for erectile dysfunction and pulmonary hypertension. These breakthroughs highlighted the clinical significance of nitric oxide. Subsequent research validated the initial claims. These studies detailed the mechanisms by which nitric oxide exerts its effects. These later investigations included the discovery of nitric oxide synthase enzymes responsible for NO production. They also illustrated the downstream signaling pathways. The use of experimental techniques, like electron paramagnetic resonance spectroscopy, chemiluminescence assay fluorescent probes, continued to refine the understanding of these The impact of Furchgott, Ignarro, and Murad's discovery spread, the work of others led to the identification of signaling molecules like carbon monoxide, or CO, and hydrogen sulfide, or H2s. These gases now also serve as critical regulators of cellular processes. Modern applications of nitric oxide in areas such as nanotechnology and drug delivery systems are being actively investigated. The development of more selective nitric oxide donors, which could target specific tissues or cells, along with a thorough comprehension of how nitric oxide interacts with other signaling pathways, remains a focus of research efforts. In conclusion, the 1998 Nobel Prize-winning discovery transformed our understanding of cell signaling and human physiology. The legacy of Furchgott, Ignarro, and Murad's work forms the bedrock of modern biomedical research and clinical practice. The future promises further insights into the complex roles of nitric oxide and, along with those discoveries, the development of new treatments for a wide range of diseases.